public health entomology would be the question right now going on in everyone's mind. Could someone be having something to share about public health entomology?
Hello. Hello.
Yes, please.
Yes, this is George.
Yes, George.
Yes. What, to my understanding, public health entomology is the study of the insects and related to human health, animals, and the environment. That is, to my understanding, as public health entomology.
Hmm, that is a good one. George, thank you. Do we have some other contribution, please?
Yeah. Hello.
Yes, please.
Yeah, this is Franco.
Yes, Franco.
Yeah, I know entomology as an study of insects, different species, and when we relate it to public health, we are referring to a study of these insects and how they cause diseases, the environment they live, and this knowledge is very important in preventing public health related disease.
Mm. Thank you, Franco. That is awesome. And now, public health entomology, as Franco has really and George have summarized it, is a subfield of medical entomology that focuses on the study of insects and their relationship with human health. When you look at public health entomology, we are thinking about identifying and monitoring potential disease carrying insects of, um, of insects, studying their biology, behavior, and we are looking at developing effective control strategies and evaluating the effectiveness of these various interventions. So, this course will provide you with a comprehensive understanding of the role of entomology in public health. So, our learning objectives for this course are to define and explain the significance of entomology in public health, to identify common disease vectors and pests of public health concern, to understand medically important arthropods' life cycle, behavior, and ecology, analyze the impact of human activities on the evolution of vector-borne diseases, evaluate strategies for vector control and management, analyze the role of entomological surveillance in disease prevention, and lastly, to apply this entomological knowledge to the public health challenges. So let's look at entomology. Uh, as still Franco has said in his submission, entomology is a scientific study of insects. It involves their behavior, evolution, ecology, anatomy, genetics, and physiology. So it is generally studying insects and everything that concerns them. So it is very important because it plays a very important role in public health due to its direct impact on the vector-borne diseases. And these vector-borne diseases, or maybe someone can tell us what they think vector-borne diseases are before we continue. This is an interactive session, so I expect you to contribute so you can follow along and have fun during the class. Anyone to tell us what vector-borne diseases are?
Yeah,
Yes.
I think vector-borne disease. These are diseases which are caused by microorganisms, which can be in the form of bacteria, fungi, viruses, and so forth.
Yeah, thank you very much. And what is your name?
Yes.
What is your
Yeah,
name?
this is still Franco.
This is still Franco. Franco, thank you so much.
Franco.
I hope all of you have got what Franco has discussed with us. And
Can I
those
give
are
a yes?
you have a submission as well. And who is this?
Yeah, this is Eguma Mikaya.
Yes.
Yeah, I appreciate the answer Franco gave. Looking at microorganism, almost every disease is being caused by microorganisms. So, vector-borne diseases, in addition to that, to me, refers to a disease whose microorganisms are being carried by other a secondary organism that serves as a reservoir then before it is being transmitted to the human. For example, mosquito is a vector that transmits malaria parasite. So a vector becomes like a secondary means of transportation of disease causing organism to human being. Thank you.
Thank you, Aguma. Wonderful submission. Thank you for the addition on top of what Franco shared with us. Vector-borne diseases are infectious diseases caused by pathogenic agents, as you mentioned, bacteria, protozoa, viruses, but these have to be transmitted by a vector. We don't have to forget that because we may be looking at something else. In this case, we are looking at those that are caused by these agents, pathogenic agents, but still are carried by a vector. That is clear. So, what is the role of medical entomologists or public health entomologists? So, these contribute to the development of different diagnostic tools, treatments, and prevention methods for these vector-borne diseases. And their work includes understanding patterns of disease exposure, developing surveillance systems, and also implementing control measures to mitigate the impact of these diseases on communities, and so many others. They also develop drugs and treatments for vector-borne diseases. They disseminate public health initiatives addressing these vector-borne disease threats. I mean, there are so many other roles that a public health entomologist does. So this course unit will be rich when you really understand it. Um, moving on, let's look at the burden of these vector-borne diseases. Vector-borne diseases, according to WHO, account for more than 17% of all infectious diseases, causing more than 700,000 deaths annually. Many of the world's most infectious diseases are transmitted by insects, and these insects rapidly spread these illnesses from one individual or host to another. WHO still estimates that one-sixth of global illnesses and disabilities is attributed to these vector-borne diseases. So the most common vector-borne diseases include dengue, malaria, trypanosomiasis, also known as sleeping sickness, yellow fever, and schistosomiasis. So, any other examples? Do you have any other examples? Vector-borne diseases? George, Franco, Eguma, let me call some other people. Some people seem to be silent. Peter, Peter,
Yes.
yeah.
Yeah, we can also talk of mosquitoes, that is female anopheles mosquitoes. We can talk also of black flies that transmitted the onchocerciasis, babulus, the OB,
Mm.
as well as many others.
Oh, George, we are looking at the diseases and not the vectors. But thank you for your
Oh,
contribution. You
all
mentioned the
right,
diseases somewhere.
all right, all right.
Peter, do you have something to tell us? Um, Gabriel, Franco.
Let me just supplement in one of the diseases that called lymphatic leishmaniasis in the simple stem.
Oh, Gabriel, we can hardly hear you. We can hardly hear you.
Okay, I'm saying
Mm.
lymphatic leishmaniasis, one of the diseases.
We can hardly hear you, Gabriel. Sorry. Let's hear from someone else. James,
Hello.
yes, Waswa.
Good evening, Madam Jessica.
Good evening. How are you?
In addition to vector-borne diseases,
Mm.
I think bilharzia also among them.
Bilharzia. Which vector causes bilharzia?
I think it is spread by water snails.
Okay. Thank you, Waswa, for the contribution. Do you have any other? Or we can go ahead. Okay. So these diseases are frequently common in tropical and subtropical climates of developing countries. Most of these diseases are listed among the 18 neglected tropical diseases, the NTDs, by WHO. So, what are neglected tropical diseases, the NTDs? These are diseases that have been always overlooked in public health efforts. And these diseases, maybe trachoma, river blindness, sleeping sickness, that is trypanosomiasis, Chagas, lymphatic filariasis, hookworms, tapeworms, among others. And these primarily affect individuals living in low-income and middle-income countries. They are rare in high-income countries, and they're often neglected due to their limited resources and lack of awareness. Most people don't know about them, and this results in inadequate funding and research. So less information is out about them. So people in low income countries suffer in silence, and therefore other funding research organizations don't offer funding, and they end up being neglected in the research area. So they're mainly prevalent in communities that are impoverished, these tropical areas, we talked about that. They are more common in South America and Sub-Saharan Africa, and they are less prevalent in Southeast Asia and Central America, those are income countries. It is estimated that NTDs affect more than 1 billion people, while the number of people that are requiring NTD interventions, that is both preventive and curative, is about 1.6 billion. That is according to WHO. This year. Many of them are vector-borne. Control of these diseases very challenging. So let's look at the dynamics of these vector-borne diseases. So looking at this diagram, it is communicating something. Using a mosquito as an example, we can see an infected mosquito, an infected host, infected mosquito, and a new uninfected host. So generally, this is how the cycle goes in simple terms. For the case of malaria, the uninfected mosquito bites the infected host, it gets infected and then transmits the plasmodium to the new infected host. And then the cycle continues. So within the system or the cycle, we have pathogens, and the pathogen is mainly the disease-causing agent. And its ability to infect, multiply, and be transmitted depends on various factors, including its virulence. We are going to talk about that. Its ability to evade host defenses and break the immune system, and the compatibility with the vector and the reservoir. We also have a vector, and these vectors are organisms such as mosquitoes. Mosquitoes, the most common one, or ticks, that transmit these pathogens from one host to another. The efficiency of transmission depends on factors such as vector competence. We are going to talk about that. And this is the ability of the vector to acquire, maintain, and transmit the pathogen, vector abundance, and then vector behavior. We also have the reservoir, and the reservoir is an organism that harbors and maintains the pathogen in nature intrinsically. So the presence and density of the reservoir population influences the likelihood of pathogen transmission in a community. So this diagram is giving us an example. Bats were prominent, very prominent, when we were talking about SARS, COVID-19, and Ebola virus. They always talk about bats as natural reservoirs of many viruses, viral diseases, flu, and then rodents. So the natural reservoirs carry the pathogens, and these pathogens are transmitted to birds, humans, animals, among others. Well, you can find time during your free time and look at this extensively. So we also have the host, which is the organism that is being attacked by the pathogen or the parasite. So let's look at some definitions, key definitions, as we look at the dynamics of vector-borne diseases. So we have the vectorial capacity, which is defined as the average number of new host infections arising from an infectious host per day.
Sorry, repeat again. Hello?
Yes, please.
I'm not getting, I didn't see the diagram and I'm not getting you very clear.
Which diagram? Didn't you see the previous diagram?
Yeah, the one we are discussing. I'm just hearing, but I don't see it.
Oh, we've already moved past it. Go back a little bit. Are you seeing it now?
I'm not seeing it.
Is anyone having the same problem?
I think the problem is with this internet we are seeing. Maybe you can get in.
Please leave and join in again.
You can continue, I'll see it.
Okay, we can be continuing for the meantime.
Okay.
So the bacterial capacity at each day is the daily number of potential infections produced by a single infectious individual. Okay? So we have different parameters that affect the bacterial capacity. We have vector competence, we have basic reproductive number, and so many that we are going to see ahead. So the basic reproductive number is the expected number of secondary cases produced by a single infection in a completely susceptible population. This basic reproductive number was commonly understood like previously during the SARS COVID-19, where every individual could infect 3 to 4 other individuals. So the higher the basic reproductive number, the more likely the disease to spread faster within the population. So vector competence determines how susceptible a vector is to transmission. So there are some important parameters that have a greater impact on bacterial capacity than others. So looking at the bacterial capacity and the basic reproductive number, they usually have a linear correlation. So the bacterial capacity and the vector population influence the productive number. So let's look at it in this case. This is the main formula of the bacterial capacity. It has various components where we see the vector density, vector biting rate, vector competence, pathogen extrinsic incubation period, and probability of the vector daily survival. So the vector density, vector capacity, and we were looking at vector density in particular, which is our aim in this case. So vector density, we're defining it as the number of that particular vector, for example, a mosquito, that is present in a particular area at any time. So vector density is usually used in public health research or epidemiological research to understand the spread of infectious diseases that are transmitted by vectors. The most common measure for vector density includes human landing rate surveys, counts of mosquitoes on human hosts, or trained animal surrogates. So, looking at vector competence, that is our B, it looks at the intrinsic ability of the vector to transmit an infectious agent. Our vector-borne biting rate, which is our A, is the number of biting incidents of mosquitoes per human population. This rate is influenced by factors such as mosquito population density, humidity, temperature, and the presence of standing water where mosquitoes breed. So some of these factors, maybe like temperature, and we know that when the temperatures are really high, mosquitoes don't really appear. Very few mosquitoes appear, but when it's really cold, mosquitoes appear, and the biting rates increase. So high vector biting rates can lead to spread of diseases like malaria, dengue, and Zika virus because the vectors are active. They're actively biting and transmitting the disease. So, giving an example of a mosquito repellent and other control measures that can be used to reduce the insects, like, for example, insecticides, mosquito nets, they can help reduce the vector population and decrease the biting rate, thereby reducing malaria spread in the community. So, we have the extrinsic incubation period, which is our N. The extrinsic incubation period is the pathogen incubation period between the time when a mosquito takes a meal, a blood meal, and the time when the mosquito becomes infectious. So the intrinsic incubation period is different from the intrinsic, that word, extrinsic and intrinsic. So intrinsic incubation rate is the time between a human being infected and the onset of symptoms due to infection. Extrinsic is for pathogen. Intrinsic is for humans. I hope that is clear. Moving on. So successful transmission depends on interactions among the components in the vector-borne system, pathogen, vector, and the reservoir, genotypes of pathogens and vectors, ecological factors, and then climate variability, and some other factors. So when we look at ecological factors, the ecological context includes habitat characteristics, land use changes, human activities, biodiversity, and this can influence the distribution and abundance of vectors and reservoirs, as well as interactions between them. So changes in these ecological conditions can alter transmission dynamics by affecting the vector and reservoir populations, habitat stability, and contact and biting rates between hosts and vectors. So when you look at human activities such as deforestation, habitat fragmentation, these play a very significant role in disease management. And looking at the mosquito, when you cut grass around the home, you are destroying the habitats of the mosquitoes. So you are actively participating in managing malaria. The structural ecological changes also increase the contact rates between these vectors and the host populations, leading to secondary epidemiological cycles, and disease emergency. So, for example, warmer temperatures may expand the geographical range of vectors, while changes in precipitation patterns can impact vector breeding sites and alter transmission risks of certain diseases. So, let's look at virulence. Virulence is a pathogen's or microorganism's ability to cause damage to a host. It's the ability of the pathogen to cause damage to a host. So high virulence corresponds to high mortality and severe illness. So diseases like malaria, Ebola, cholera, SARS, COVID-19, HIV have a very high virulence. So virulence is different from pathogenicity, because pathogenicity of an organism is its ability to cause disease. It's determined by the virulence. So the ability to damage the host is different from the ability to cause disease. So the diseases that have high virulence always have an increased reproduction within the host. And then the low virulence diseases result in low or no mortality and mild symptoms. And the examples of these are common cold. So these diseases can easily be treated with rest, fluids, or over-the-counter medication. These have mild symptoms. However, it is very important to note that the low virulence does not mean that these diseases should be taken for granted, as they still can cause discomfort and impact on daily life, on the daily health of the host. So these diagrams are giving us some view on virulence, and we see this patient has a very high virulent disease, and this person is in bed already sleeping, so they have less chance for transmission. And these ones that are less virulent, they have more chances of transmission because they don't really put you down and you're transmitting them really fast. So let's compare like HIV, AIDS, and flu versus and flu. HIV/AIDS spreads a little lower because it really puts you down as compared to common flu. So let's move on to pathogen evolution. When you talk about pathogen evolution, what comes to your mind? Going back to, let me look for some names here. To Eguma.
Yes.
When you talk about pathogen evolution, what comes to your mind?
What comes into my mind is how pathogen has been discovered. I just think about the discovery of pathogen, how disease-causing organisms have been discovered.
Okay, thank you. Looking out for other names. James, you haven't said anything. Oja James, do you want to tell us something? Alex.
Yes.
James, go on.
No.
Oh yeah, Dennis, go on.
This is Dennis. Yeah. To me, when we talk of pathogens evolution, we meant how the bacteria evolves from being non-pathogen to becoming pathogenic. In other words, how they colonize the host and they become pathogenic in nature. Thank you.
Okay. Good. Michael, do you have something to add? Kodika Michael? Okay, Michael doesn't have. We have Alphonse. Alphonse, do you have anything to tell us? No. Franco, do you have anything for us? Franco, are you there?
Yeah, hello.
Yes, Franco. Do you have something for us?
Yeah, thank you for giving me another chance. I don't have much to add on what the other colleague has said, but I think basically this involves how pathogens changes over time. You know, sometimes maybe the genetical makeup and all these other structures change with time. That is what I can add. Thank you.
Thank you very much for all your contributions. So, pathogen evolution refers to changes in genetic makeup of pathogens over time in response to the selective pressures created by the immune systems of the hosts and the vectors, vaccine deployment, antimicrobial drugs, and so many other interventions. So the pathogens keep evolving and changing the genetic makeup with time. So the evolutionary theory suggests that organisms aim to maximize reproduction, potentially leading to higher virulence. These pathogens really evolve to beat these interventions so that they can maximize their transmission rates. So pathogens' reproductive success depends on the ability of its offspring to find new hosts, which equates to transmission success. High virulence increases transmission probability through contact and infection of new hosts. So organisms employ various reproductive strategies to maximize their chances of reproducing successfully. So this figure here is giving us a view of an idea of evolution. But you're looking at a mosquito population that is ancestral, and then the evolved population that has undergone multiple generations of coevolutions within within its population. So these mosquitoes, let's concentrate on the pathogen load. You see that the pathogen load is smaller in the ancestral population as compared to that in the evolved population. So when the mosquitoes become more tolerant to to the drugs administered to them, the pathogens keep growing within them and they do not get destroyed. So their number increases, the pathogens within the evolved population is really higher compared to that in the ancestral population. So when we are looking at public health strategies to curb malaria, we have to also consider the evolving genetic or the changing genetic makeup of the mosquitoes. And I think that is microbiome studies, like where people really concentrate on the studying the genetic makeup of the mosquitoes or the microorganisms as they change over time so that you can have targeted evidence-based interventions to control these infectious diseases. So high pathogen density in the host's bloodstream increases the likelihood of mosquito infections during a bad meal. And however, excessively sick hosts may not spread infections effectively due to reduced activity that we saw in the other patient who was sleeping. And so a balance is required. We need enough pathogens to infect new hosts, but not enough to incapacitate the host entirely, because we need that host to also transmit the what? The disease. So transmission models leading to high violence include vector-borne diseases like malaria that we already talked about. So pathogens do not need hosts survive for transmission to occur as long as new hosts encounter the pathogens before it dies. So the major goal is to reduce the transmission probability of pathogens in public health. So let's look at something else lastly, the ARA strategists and the case strategists. So these are just contrasting reproductive strategies adopted by organisms in response to environmental conditions. So we have an ARA to K scale of the reproductive strategy, where the organisms are balancing between the egg output and parental care. So the organisms that really don't give much parental care to their offsprings usually don't survive for long. So they usually have a lot of they lay maybe a lot of eggs. For example, ticks lay really so many eggs, but they do not care about them. So the ticks that grow are smaller. And when you look at this, when you look at the oysters as an example for the ARA strategy, because this scale is moving from ARA to K, you can see that the oysters produce 500 million fertilized eggs a year, but they have no parental care. Hmm? The frog produces 200 eggs per year, but it doesn't have parental care. When you look at the gorilla, it has one baby every 5 years and it takes care of the baby. So when we look at the R and K strategy, we are looking at balancing between egg output and parental care. So let's look at them in a little bit more detail. The K strategists have intense competition in crowded environments. They have fewer offsprings. They invest more in each offspring to enhance their ability to compete. That is, they offer parental care. And they have a larger body size and longer generation time. They tend to have a stable population close to the carrying capacity of their environment. And examples are humans, elephants, whales, and so many others. Most of the animals actually fall under this case strategist category. Yeah, those are some of the examples of the case selected species. So looking at the ARA strategists, they are relatively smaller organisms with early maturity and a short generation time. They have very many offsprings, they lay very many eggs, but they are capable of dispersing their offspring widely. They have little or no parental care at all, and they have less competitions within their species. So survival is influenced by luck, and they tend to have a population boom and bust cycle with rapid population growth followed by a crash. So most of the examples include insects, mice, and many fish species. And that is giving us some of the aerated species. I think that is the end of my presentation for the introduction to entomology. Public health entomology today. Thank you so much for listening.